Team number 5, Enterobacteria, Escherichia, Shigella, and Salmonella. Plan of the team. Morphology and identification: gram-negative, non-spore-forming rods, catalase positive, oxidase negative, and primarily normal flora of gastrointestinal tract, transient colonizers of skin, water streams, and facultative anaerobes, mixed acid fermentation. All ferment glucose, all reduce nitrates to nitrites, and lactose fermentation, usually normal flora positive and pathogens negative. Primary isolation media include eosin methylene blue and McConkie agar, and differential selective media for specific organisms include Salmonella Shigella media and bismuth sulfate media and Hecton enteric agar, XLD and deoxycholate citrate agar. Enterobacteriaceae are divided ferment lactose fermenting and non-lactose fermenting. Lactose fermenting are divided Escherichia coli and Klebsiella pneumoniae, and non-lactose fermenting groups motile and H2S producing, and non-motile and non-H2S producing. Proteus species and Salmonella species, Shigella species, Yersinia pestis, and Yersinia pseudotuberculosis. Antigenic structure: most are motile by Peritrichus, flagella, H antigen and capsule, K antigen, V for Salmonella, lipopolysaccharide, endotoxin, O antigen, and various outer membrane proteins, pili, various antigen types, some encoded by plasmids. O antigen is lipopolysaccharide, H antigen in flagella, and K antigen capsular antigen. And structure of bacteria: flagella H antigen and in capsule K antigen, and endotoxin or lipopolysaccharide, and outer membrane, periplasmic space, lipoprotein and peptidoglycan, in the membrane, cytoplasm, pili. Enterobacteriaceae foodborne infection: Escherichia coli, Salmonella, and Shigella. Escherichia coli, it is also the most common aerobe to be harbored in the gut of humans and animals. After excreted in feces, it remains only for some days in the environment. Hence, detection of Escherichia coli, especially a variant called thermotolerant Escherichia coli, survives at 44 degrees Celsius, is taken as an indicator of recent contamination of drinking water with human or animal feces. Other species are less important as human pathogens. Escherichia coli toxins: two types of enterotoxin, Shiga type toxin and heat-stable toxin, hemolysins, and endotoxin, heat-label toxin, and enterotoxins. They are produced by diarrheogenic strains of Escherichia coli. They are of three types: heat-stable toxin, heat-label toxin, and verocytotoxin. All have been and the hemolysins. They are produced more commonly by virulent strains of Escherichia coli, especially pyelonephritis strains. However, their role in pathogenesis is unclear. And the cytotoxic necrotizing factor 1 are secreted autotransporter toxin. They are cytotoxic to bladder and kidney cells. And the adhesions: colonization factors, both pili or fimbrial, non-fimbrial factors involved in attachment. There are at least 21 different types of adhesions. And febrile antigen pilus is the organ of adhesion, helps in attachment and colonization. It expressed by a few strains of Escherichia coli. Various febrile antigens are colonization factor antigen. It is a type of febrile expressed by enterotoxigenic Escherichia coli and mannose resistant febrile. They hemagglutinate with RBCs that is not inhibited by mannose. These are expressed by uropathogenic Escherichia coli and helps in colonization of these strains onto uroepithelial cells. And the P. febrile binds specially to the P blood group antigens present on human RBCs and uroepithelial cells. And virulence factors that protect the bacteria from host defenses, capsule or iron capturing ability, automembrane proteins. Somatic O antigen. It is a lipopolysaccharide antigen. It is heat stable. Occasionally it cross-reacts with O antigen of other species, and early O serotypes are usually the commensals of intestine. Late O serotypes are the diarrhea-producing strains, and serotyping is done by slight agglutination with specific O antisera. O antigen is the most important virulence factor responsible for endotoxic activity. It protects the bacteria from phagocytosis and bactericidal effect of complement. Flagellar or H antigen. It is heat label. Presence of H antigen flagella makes the bacteria motile, hence contributing to their virulence. Capsular K antigen. It is a polysaccharide capsular antigen present on the envelope or microcapsule of a few strains of Escherichia coli. When present, it encloses the O antigen and renders the strains inagglutinable by the O antiserum. It may also contribute to virulence by inhibiting phagocytosis. It is expressed by only few strains of Escherichia coli, thus causing neonatal meningitis, pyelonephritis, and septicemia. Enteropathogenic Escherichia coli, infant diarrhea, fever, vomiting and nausea, non-bloodly stool, destruction of surface microvilli. This is a problem mainly in hospitalized infants and in the care centers. And enteropathogenic Escherichia coli causes infant diarrhea and occasionally causes sporadic diarrhea in adults, and the person-to-person spread is seen. And and next is it is enteropathogenic Escherichia coli. It is non-toxigenic and non-invasive. Mechanism of diarrhea: adhesion to intestinal mucosa mediated by plasmid-coated bundle-forming pili, which form cup-like projections called pedestals. And these attaching and efficient lesions. These are typical lesions produced on the intestinal epithelium, which leads to disruption of brush border epithelium, causing increased secretion and watery diarrhea. Next is enterotoxigenic Escherichia coli. Is the most common cause of traveler's diarrhea, causing 75% of cases. It causes acute watery diarrhea in infants and adults. Common serotypes associated are O6, O8, O15, O25, O27, 153, and 159, and etc. It is toxigenic but not invasive. Pathogenesis of enterotoxigenic Escherichia coli is by attachment to intestinal mucosa, is mediated by febrile protein called colonization factor antigen, and toxin production, heat label toxin or heat stable toxin and heat label toxin. And diagnosis is done by detection of toxins by in vitro and in vivo methods. Transmission is via contaminated food or water. Enterotoxigenic Escherichia coli causes diarrhea like cholera but milder, caused by these toxins and colonization factors. This slide shows heat stable toxin and heat stable toxins influence. And interinvasive Escherichia coli. The organism attaches to the intestinal mucosa via pili automembrane proteins are involved in direct penetration, invasion of the intestinal cells, and destruction of the intestinal mucosa. This is lateral movement of the organism from one cell to adjacent cells. Symptoms include fever, severe abdominal cramps, and watery diarrhea, followed by bloody diarrhea, mucosa and pus present. And common serotypes associated with enterinvasive Escherichia coli are O28, O112, and 114, 124, 136, and 152. Pathogenesis enterinvasive Escherichia coli is not toxigenic but invasive. The epithelial cell invasion is mediated by a plasmid-coded antigen called Virulence maker antigen. And the enterinvasive Escherichia coli is biochemically, genetically, and pathogenically closely related to Shigella. And the manifestations this include ulceration of bowel, dysentery, diarrhea with mucus and blood, called bacillary dysentery resembling Shigellosis. Diagnosis, detection of Virulence maker antigen by ELISA, and HeLa cell invasion assay, and inoculation of bacterial suspension into guinea pig iris production conjunctivitis, and compared with other Escherichia coli strains. And enterinvasive Escherichia coli are biochemically atypical, being non-motile and lactose non-fermenters, and negative for lysine decarboxylase. And next, enterinvasive Escherichia coli dysentery resembles Shigellosis. The enterinvasive Escherichia coli are like Shigella but non-motile and non-lactose fermenter. And next, enterohemorrhagic Escherichia coli. Serotypes associated with enterohemorrhagic Escherichia coli are O6, O55, O91, 103, and 111, and 113. And enterohemorrhagic Escherichia coli is usually transmitted by contaminated food, consumption of spinach and sprouts and undercooked ground beef. It is prevalent mainly in industrialized countries, in contrast to other diarrhogenic Escherichia coli, which are common in developing regions, and low infective dose. The infective dose of enterohemorrhagic Escherichia coli is very low. Only few organisms are required to initiate the infection. Pathogenesis of enterohemorrhagic Escherichia coli secretes a toxin called verocytotoxin or Shigella-like toxin. Shigella-like toxin mechanism of action. Shigella-like toxin acts by inhibiting the protein synthesis by inhibiting the 28S subunit of 60S ribosome. Shigella-like toxin is of two types, STX1 and STX2. The STX1 is indistinguishable from Shiga toxin produced by Shigella dysentery type 1, and the STX2 is more commonly associated with development of hemorrhagic uremic syndrome than STX1. Enterohemorrhagic Escherichia coli, hemorrhagic colitis develops and bloodly and copious diarrhea, few leukocytes, and afebrile and hemolytic uremic syndrome, hemolytic anemia, and low platelets, and kidney failure. Enter-aggregative Escherichia coli, a cause of persistent watery diarrhea with vomiting and dehydration in infants. The bacteria adheres to the intestinal mucosa and elaborates enterotoxins, heat-stable toxin. The result is mucosal damage, secretion of large amounts of mucus, and secretory diarrhea. Enter-aggregative Escherichia coli, it is so named because it adheres to HEP2 cells in a distinct patterned layering of the bacteria aggregated in a stacked brick fashion. For most strains are untypable but H-typable. Pathogenesis, intestinal colonization, is mediated by aggregative adhesion fimbriae. It also produced enter-aggregative heat-stable enterotoxin. 1. Manifestations, persistent and acute diarrhea, are commonly seen, especially in developing countries. It is an enter-aggregative strain that has caused major outbreaks in Germany in 2011. One peculiar feature of this strain is it produces Shigella-like toxin and can cause of hemorrhagic uremic syndrome. This slide shows main characteristics of each type of Escherichia coli. And the next group of enterobacteria is Shigella. Shigella is the most important agent of bacillary dysentery. Is named after Japanese microbiologist Kiyoshi Shiga, who isolated the first member, Shigella dysentery serotype 1, in 1896 from epidemic dysentery. Shigella differ from Escherichia coli being non-motile and non-fermenting. Most sugars except mannitol, and classification based on a combination of biochemical and serological characteristics. Shigella are classified into 4 species: Shigella dysentery, Shigella flexneri, Shigella botte, and Shigella zonae, which are also designated as serogroups A, B, C, and D respectively. General features contain pili. Most strains cannot ferment lactose. Shigella zonae can slowly ferment lactose according to O antigen for serogroups, easily acquiring drug resistance. Shigellosis, within 3 days, epithelial cell damage. Shigella is one of the important cause of bacillary dysentery. Mode of transmission: infection occurs by ingestion through contaminated fingers, most common food and water, or flies. It can also be transmitted sexually, and the minimum effective dose as low as 100 bacilli are capable of initiating the disease, probably because of their ability to survive in gastric acidity. Entry via M cell. Bacilli enter the mucosa via M cells. They cross the basolateral side of M cells to reach the submucosa, where they are engulfed by macrophages. Subsequently, the macrophages release bacilli. Cytokines are released by infected intestinal epithelial cells, which attract increased numbers of inflammatory cells to the infected site, and inflammation, and leading to the acute colitis that characterizes shigellosis. invasion. Once inside the submucosa, Shigella induce their own uptake into the adjacent epithelial cells. Invasion is determined by a large virulence plasmid, which codes for important virulence factors such as EPA proteins and the type 3 secretion systems. They in turn help in uptake of the Shigella into the host cell. Direct cell-to-cell spread. Shigella spread directly from one host cell to the other by inducing actin polymerization of host cell mediated by ECSA proteins. Action filaments attach to one pole of the bacilli and form a tail. This helps in propelling the bacilli through the cytoplasm. Once the organisms come into contact with host cell membrane, cellular protrusions are formed and the bacilli are engulfed by neighboring cells. Shigatoxin, cytotoxic, enterotoxic, and inhibits protein synthesis. Lysine 28S RNA. Incubation period lasts from 1 to 4 days. After the incubation period, fever, cramping, abdominal pain, and watery diarrhea. For 3 days. This may be followed by frequent scant stools with blood, mucus, and pus due to invasion of intestinal mucosa. It is rare for the organism to disseminate. The severity of this disease depends upon the species. Salmonella dysentery is the most pathogenic, followed by Flexneri, Zone, and body. Clinical significance: men only. The reservoir transmission fecal to oral contact, insects, and transmitted by adult food handlers, unwashed hands. Diagnosis of Shigella. Diagnosis of Shigella infection. Specimen: fresh stool, mucus flex, and rectal swabs or serum to demonstrate rise in titer of agglutination antibodies. Selective media: Hecton, enteric agar, and Shigella salmonella agar, and Shigella salmonella agar, and xylose lysine deoxycholate medium, and deoxycholate citrate agar. And identification: slight agglutination. Slight agglutination test. Because of biochemical variations, identification of Shigella is always confirmed by slight agglutination with polyvalent antisera. Then the species identification can be done by using group-specific antisera specific for serogroups A, B, C, or D. Serotypes under each species are further detected by using type-specific antisera. And culture smear and motility testing. Gram stain of colonies reveal short gram-negative bacilli measuring 1.5 millimeters, and they are non-motile, non-capsulated, and non-sporing. Colonies of Shigella appear red without black center. It is superior of deoxycholate citrate agar, as it is less inhibitory to Salmonella dysentery and Shigella flexneri. This slide shows main characteristics of Shigella species and virulence factors of Shigella. Next type of enterobacteria is Salmonella. Salmonellosis may present as one of the several syndromes, including gastroenteritis, enteric typhoid fever, or septicemia. Salmonella is antigenically complex. The classification of nomenclature of Salmonella have undergone several modifications over the past years. There are several classifications proposed so far. It is the oldest user-friendly classification which is still widely used. It divides Salmonella into two groups: typhoidal Salmonella and non-typhoidal Salmonella. Typhoidal Salmonella, it includes serotypes Salmonella typhi and Salmonella paratyphi. They are restricted to human hosts in whom they cause enteric fever, typhoid or paratyphoid fever. Non-typhoidal Salmonella, the remaining serotypes can colonize the intestine of a broad range of animals, including mammals, reptiles, birds, and insects. They also infect humans, causing foodborne gastroenteritis and septicemia. Virulence factors. Endotoxin may play a role of intracellular survival. Capsule for Salmonella typhi and some strains of Salmonella paratyphi. Adhesions both febrile and non-motile. Type 3 secretion systems and effector molecules. Two different systems may be found. One type is involved in promoting entry into intestinal epithelial cells, and the other type is involved in the ability of Salmonella to survive inside macrophages. Outer membrane proteins involved in the ability of Salmonella to survive inside macrophages. Flagella helps bacteria to move through intestinal mucus. Enterotoxin may be involved in gastroenteritis and iron capturing ability. And enteric or typhoid fever. Enteric or typhoid fever occurs when the bacteria leave the intestine and multiply within cells of the reticular endothelial cells. The bacteria then re-enter the intestine, causing gastrointestinal symptoms. And typhoid fever has a 14 days incubation period and may last for several weeks. Salmonella typhi is the most common species isolated from this type of salmonellosis. Human reservoir carrier state common. Contaminated food, water supply, poor sanitary conditions. V antigen is the surface polysaccharide envelope or capsular antigen covering the O antigen. The naming is due to the belief that V antigen is related to virulence. It is expressed in only a few serotypes such as Salmonella typhi and Salmonella paratyphi. And when V antigen is present, it renders the bacilli inagglutinable with the O antiserum. However, the strain becomes agglutinable after boiling or heating at 100 degrees Celsius for 1 hour, which removes V antigen and exposes the O antigen. V antigen is also destroyed by HCl and alcohol and other disinfectants. As V antigen is purely immunogenic and the antibody titers are low, it is not helpful in the diagnosis of cases. The V antigen is not employed in the vital test. However, it is believed that the complete absence of the V antibody in a proven case of typhoid fever indicates poor prognosis. Salmonella gastroenteritis. Salmonella gastroenteritis is the most common form of salmonellosis and generally requires from an 8 to 48 hours incubation period and may last from 5 days. Symptoms include nausea, vomiting, and diarrhea, no bloodly stool, and the Salmonella enteritis is the most common isolate. Portly eggs, no human reservoir, self-limiting 5 days. Salmonella septicemia or bacteremia may caused by any species, but Salmonella Shigellosis is the most common. This disease resembles other gram-negative septicemias and is characterized by a high remittent fever with little gastrointestinal involved. Typhoidal salmonella include Salmonella typhi and Salmonella paratyphi A, B, C, which cause enteric fever pathogenesis. Salmonella are transmitted by oral route through contaminated food or water. Infective dose of Salmonella is higher than that of Shigella. Minimum 10 bacilli are needed to initiate the infection, and risk factors that promote transmission include the conditions that disease, stomach acidity and intestinal integrity, inflammatory bowel disease, and gastrointestinal tract surgery or suppression of the intestinal flora by antibiotics. Entry through epithelial cells lining the intestinal mucosa. Salmonella can trigger the formation of membrane reflux on the cell membrane of M cells. These reflux reach out includes the adherent bacteria within the large vesicles. This process of uptake is called bacteria-mediated endocytosis. And diagnoses the samples, specimens, enteric fever, blood, bone marrow, stool, urine. At gastroenteritis, we take stool, vomit, suspected food, and at septicemia, we need blood. And culture isolation, blood and bone marrow culture. In first week of illness, in blood culture bottle, brain heart infusion broth and automated BACTEC, and stool culture 4 weeks in 4 weeks of illness. Enrichment broth such as selenite F broth and the gram-negative broth. Low selective media, McConkie-Agar, transolent NLF colonies, and highly selective media. XLD agar and Shigella Salmonella agar, and Wilson Blair's bismuth sulfate media, and the urine culture done on McConkie agar and culture smear and the motility motile gram-negative bacilli. Biochemical identification: catalase positive and oxidase negative. Nitrate is reduced to nitrite, and slight agglutination test to confirm the serotype, and this culture method. This slide shows what type of medium we use for cultivation of Salmonella. This slide shows main characteristics of Salmonella pathogen and types of Salmonella present in this slide. Thank you very much.